About the semi-conservative nature of DNA replication. Now, we will not be touching up on the semi-conservative manner of this segment yet. That will be in the end of it. Right now, we're going to see how we can duplicate this DNA molecule to produce a new one. Now, this is our starting DNA molecule, and if you are paying attention, which of course you are, you'll clearly see that I have labeled one strand in one color and the other strand seems to be in another color. Just to prevent even further confusion, I'll color them completely separately. Yeah, something like that. So the bases are in the middle. It's a different color. That's not that relevant. Now, I'll start off with an introductory point. DNA replication. If you remember, if you recall back the cell cycle, DNA replication occurs during the S phase. The S phase is part of interphase, if you remember. Interphase of the cell cycle. Let's do that. The process is regulated by a series of enzymes, just like any other biological process in the body. We will use this piece of DNA as our base or starting point to actually talk about how DNA replication occurs, how it begins. Now, first of all, here's the first and foremost thing that I want to tell you. You cannot replicate or duplicate DNA like this. DNA is a double-stranded helix. We all know that. But here's the problem with this helix. This helix cannot be replicated, cannot be duplicated like this. You need to first open things up. Now, if you look at this helix, how do you open up the two strands? How do you separate these two strands? That would be to break these. What are these? These are hydrogen bonds. Breaking hydrogen bonds between the nucleotide bases is not that hard of a deal. For that, we have this guy. I'll do my best to draw a nice little triangle right here. So this guy, you see, he's pointy. So he can go and break. Boom, 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 boom. His name is helicase. You can call him DNA helicase, but you can just call him helicase as well. So he's going to go over and destroy the bonds and unwind the double helix. Now, what would that look like? Let's go down. So pointy arrow down. And here's what the exposed strand after the unwinding looks like. So the first coil has been unwound, and therefore we have a second strand here as well. I will leave it like that. Yes, like so. And here's the helicase going further. Like so. He's destroying the rest of the hydrogen bonds this way within this path and just uncoiling the DNA because we need to separate it first. Now let's pull our focus from this part and onwards to this. From this point onwards, you will only be focusing on this. So let me bring that down and let me also do something like this just to extend it a little bit. Okay, cool. The enzyme helicase has successfully unwound the DNA double helix by breaking the hydrogen bonds between the nitrogenous bases. Do you have to remember all of those verbal things? No, I'm going to give this in point form later. But first of all, we have to understand the process that is going on, like so. Now, in doing so, you can clearly see we have two strands. One of these strands is known as the leading strand. One of these strands is known as the leading strand, and then the other one is known as the lagging strand. Known as the lagging strand. Now, the only difference is the leading strand runs from 3' to 5'. What on earth is that? That's not new to you. If you refer to DNA structure, the previous lecture series, you will understand what this 3' and 5' business is. Let me center this. Okay. And the lagging strand runs from the opposite direction. We know DNA is an anti-parallel molecule. One runs in one direction, the other runs in the other direction. So therefore, this is the structure we have. After this DNA is unbound, this structure is given a formal name. This is known as a replication fork. A DNA replication fork. DNA replication fork. Okay. Step 1, what happened? DNA helicase. I'm going to write that down again. So this is, nope, not enough space. This is DNA helicase, like so. That's going to break the hydrogen bonds. And now, okay, DNA helicase has broken the hydrogen bonds, but he's moving in this direction. So can this DNA reform its coils or loops again? The answer is yes, because helicase is moving down that direction. Once he's paused through here, there's nothing holding these DNA strands from coming back together again and forming another double helix. For that, we have a very special enzyme that wraps like this around this forming replication fork. Like so. This guy is called topoisomerase. This guy is known as topoisomerase. Helicase and topoisomerase are the first enzymes to run through this process. Now you have to clearly understand how the leading strand works, how the lagging strand works, and why this distinction is given to us. Right, now let's actually begin the process of DNA replication. So for that, I'm going to introduce an enzyme. This is the next enzyme that will help us to replicate DNA. If you would just allow me to color this. Okay, done. This is primase. This is an enzyme known as primase. What primase does is, now here, whenever DNA uncoils, we have to have a specific point to begin the replication. So let's say we want to begin replication from here. We don't want these parts. We just want to begin replication from here. So we have to have a starting point now, and that is when primase comes into play. What primase does is it binds to the DNA strand and produces these little parts known as primers. These are primers. Primase produces primers. These are simply short pieces of RNA. Short pieces of RNA to mark the starting point of replication. Now if I observe the strand, I'm going to find what? I will find bases. In both of these strands, I'm going to find bases. That's your code. That's your genetic code. Only this code can transfer into your proteins and all the other information. So once primase binds, it will indicate the formation of an RNA primer and it binding to that subsequent relevant base. And a couple of these will be coiled up like this. Just a very short piece. And then you call this a RNA primer. This is a primer. This is a primer. This will mark the starting point of DNA replication. I think I should also make primase into this green. Yeah, that's better. So that is primase. Let's also label it in that particular green. Nice. This is DNA primase. You don't have to call it DNA primase. It's simply primase. What does primase do? Primase produces short RNA primers. These are short RNA primers. I'm going to get rid of that word now. I'll be giving that to you later. But remember, these are RNA molecules, not DNA molecules. These mark the starting point of DNA replication. And then only comes in the most important enzyme in this entire cascade. I'm just considering what color I should use to represent him. Yeah, I think that's good. In comes this guy. Also, please do hold while I color him. Color. This is the next, and this is the most important enzyme of this entire replication fork that is going to actually allow DNA to be replicated. Okay, done coloring. This guy's name is DNA polymerase. DNA polymerase. DNA polymerase. After primase is done with its job, DNA polymerase will come into the picture. Now it knows where to bind. Previously, DNA polymerase has no idea where to bind. Do I bind here? Do I bind here? Something like that. Now don't focus on the lagging strand yet. Focus on the leading strand. I'll talk about the leading strand first, and then I'll move on to the lagging strand because there's a small difference. It's not a major difference, it's a very small difference. Just focus on the leading strand. Okay, now this guy is going to know, oh, I have to bind here because this is where the primers are situated. This is where DNA primase, my friend, has laid down the primers. This is where I should go and bind. So he's going to bind like this. I'll just do a Control Z to get him like this. Okay, he's going to bind like this, and then he's going to go down this way. DNA polymerase is going to go down this way. While it's going down that way, it will be adding nucleotide bases, DNA bases. The primers will be replaced. Primers was just a method of showing the starting position of DNA replication. Now DNA polymerase is going to keep adding these free nucleotides, DNA bases, like so, all the way down the leading strand. It will be reading one codon at a time. We'll talk about what codons are as well. It'll be reading, and then just replicate. It's not really going to read codons because this is not protein synthesis. And if you don't understand the difference, that's okay. Protein synthesis would be the next part of this chapter. But just so you know, it's reading a base. For example, if this is A, remember adenine pairs with thymine. Simply DNA polymerase will add T to it. DNA polymerase will add T to it. That's the idea. I'll just get rid of those bases like so. Now the thing is, this direction to which DNA polymerase builds is very important. I'm going to get rid of this primers thing. I'll just put those primers here just so we don't forget about them. Primers, like so. Right, now you see this strand. This is the most important part you have to understand, otherwise the next part is not going to make sense. This leading strand runs from the 3' to the 5' direction. Leading strand runs from the 3' to the 5' direction. DNA polymerase is only capable of adding nucleotides in the 5' to 3' direction. What does that mean? Meaning, when this happens, It's building in this direction. So it's building from the 5' to the 3' end. So this is being built, then you get this, then you get this in this direction. DNA polymerase can only add nucleotide bases in the 5' to 3' direction. Always remember, that's the only direction that DNA polymerase can build. It's the only direction that DNA polymerase can build in the 5' to the 3' direction. So when it comes to the leading strand, it's not a problem. It's not a problem at all. The leading strand can be continuously synthesized because that direction is fine. This is the 5' end, this is the 3' end. So DNA polymerase just can build in the 5' to 3' direction. It's very important to understand this distinction. Do you see? The strand is running from 3' to 5'. DNA polymerase can only build in the complementary manner, meaning in the 5' to 3' direction, like so. So because of that, this will be the direction that DNA polymerase can build the strand. So leading strand again is continuously made by condensation reactions between the deoxyribose sugars and phosphate groups. Not a problem. Now the problem arises when we move on to the lagging strand. Let me pick this color. Okay. The problem arises when we move on to the lagging strand. Now, if DNA polymerase can only, I'll just replicate him, can only move in the 5' to 3' direction, for this strand, he is only going to be able to do something like this. For the above strand, he can do this continuously, build, 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 build. But here, he has to go like this. He has to go like this in this direction. And then the strand is going, and then he's going to have to jump and then build again. Because you see, he's only capable of building in the 5' to the 3' direction. He can't go the other way. Now, you can't go like this. He's not capable of adding nucleotides in that manner. He's only capable of adding nucleotides in the opposite manner. So he has to go a little bit this and then jump. Then, because you see the strand is moving, the strand is moving forward. So he's only going to be capable of adding a few nucleotides every single time until the strand moves forward, like so. And in doing so, do you see something happens? Some bases have no time to be put. Some nucleotide bases have no time to be replicated. Right? Because of that, we're going to have spaces. We're going to have holes in the lagging strand. And that is why this is known as the lagging strand. It's lagging. We have empty spaces. We have openings that we have to later fix. So however, on the lagging strand, DNA polymerase can only build in small fragments. These are small fragments. Now the thing is, these small fragments are given a specific name. These small fragments are known as, it's a very unique name. It's actually the name of a person known as Okazaki fragments. I'm going to try to see if I remember his full name. I think he was T something, Tsuneka Okazaki or something by him. I am so sorry if I'm pronouncing that name wrong. You can just give it a Google search to find his name. And he was the one who discovered that the lagging strand was only built in small portions or small fragments known as Okazaki fragments. Like so. Alright, now there's a problem. Leading strand is fine. We can continuously synthesize the leading strand without any gaps, without any issues. But here we have gaps. Who's going to help us fill these gaps? To fill these gaps, we have another special enzyme. He is known as DNA ligase. You can think of him like glue. Whoops, it's not a G, that looks like an S. Ligase. He's going to piece together these pieces like glue. is going to glue these lagging Okazaki fragments together. He's going to glue these lagging Okazaki fragments together. Let's write this procedure down in some good point-wise context. Now, the first part is let's actually use numbers. I think that's going to be better. Now, the enzyme helicase unwinds DNA double helix, unwinds the DNA double helix, the DNA double helix by breaking, let's expand that, the hydrogen bonds. So I'll call it H bonds for short. The hydrogen bonds between the nitrogenous, between the nitrogenous bases. That's the first one. In doing so, we brought life to two strands. The leading strand runs from 3' to 5'. The lagging strand runs from 5' to 3'. 5' to 3'. Okay. Part 2, the enzyme, I'll just call it easy. Oops. Topoisomerase prevents supercoiling again. Now, don't worry about what supercoiling is that much. You just think of it as the DNA coiling back onto itself like this. Okay, helicase unwinds it by breaking the hydrogen bonds, but these hydrogen bonds can be reformed again. So to prevent that, we have DNA topoisomerase. Topoisomerases are a very complex group of enzymes. We have many kinds of topoisomerases, and they slightly differ in the ever so function that they provide to this DNA molecule, but that is completely out of context for your syllabus. All you have to know is that topoisomerase is going to prevent the DNA from coiling again. That's all. Okay, now let's begin with the first enzyme that's going to be involved in the actual replication process itself. That is going to be DNA primase. Now, DNA primase, you don't even have to call it DNA primase. Let's just call it the enzyme primase. The enzyme primase synthesizes short pieces of RNA. These are known as primers to mark the starting point of replication. Now onto the big guy, DNA polymerase. He is going to add nucleotide bases in this is the most important part, 5' to 3' direction. Now, a small disclaimer. You might be confused about this 5' to 3' direction, 3' to 5'. You just take out a paper, a small A4 sheet or something, might be maybe even a whiteboard, and you just draw the leading strand, and then you see how DNA polymerase builds in the 5' to 3' direction. That's going to give you some context if you don't get it in one go. This direction is the only direction that DNA polymerase can build in. Therefore, the leading strand is continuously built. Forget the spelling mistake, continuously built, while the lagging strand is made in short segments called Okazaki fragments. Continuously by condensation reactions between the deoxyribose sugars and the adjacent phosphate groups. The leading strand is built continuously, but the lagging strand is built in these small segments called Okazaki fragments. Okazaki fragment. So I'm going to add something here to actually show you how these things are built, continuously built by condensation reactions between the deoxyribose sugars and adjacent phosphate groups. While the lagging strand is made between in Okazaki fragment. Let's actually do something like this. Hold on. Let's move that to a completely new point. While the lagging strand is made in segments, not segments, it's segments, called Okazaki fragments. Okay, DNA polymerase can continuously build that like the leading strand. Right now, we need a way to clear up these holes and this mess. So because of that, we have the enzyme DNA ligase to join these lagging segments together. Join these lagging segments together like so. This is the entire DNA replication process according to what you should know. There's of course a ton more information that has to go into this, but this is the overall picture. 2, 3, 4, 5, a 6-step process that you should know. The key thing you should pay attention to here would always be the names of the enzymes. Let's highlight the enzymes we have: DNA helicase, topoisomerase, DNA polymerase, and the final enzyme would be DNA ligase. You have to always remember, whenever it comes to biological processes, enzymes are heavily involved. Enzymes are heavily involved. Okay. Back to the point I told you that I would explain further. What do you mean by semi-conservative? Why is DNA replication known as a semi-conservative process? Why is DNA replication known as a semi-conservative process? Let's see. It's very easy to understand. Now, let's first take my first DNA molecule. Right? That's my first DNA. Actually, I'm going to get rid of those bases as well. We don't want any forms of confusion. Let's get rid of it. Write it again. Okay, boom. Like so. Now this is one DNA molecule, right? This is coiled like this, right? Now when DNA polymerase adds the nucleotides, it's going to build one like that and then the next one like this. Okay. And then these two guys are going to be a separate strand, and these two guys are going to be a separate strand. So they will coil up like this. And then they will coil up like this. Okay. Now let me get rid of those things. Let's pull them back here. Okay, like so. In orange, we have the new strands that we synthesize. The blue strand and the reddish strand were old ones. Old strands. Do you see why we call this semi-conservative? Semi means partially or half. What does that mean? Every single time that DNA replicates, half of it is retained. You see here, this is the original DNA molecule, these two colors. In the new cloned DNAs, one half of that is conserved. A new strand is formed, in each case the orange color, and the old strands are still in the DNA impact itself. DNA impact itself. That's the idea of the semi-conservative nature of DNA replication. Every single time DNA replicates, it will have one half of its original DNA strand.